Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about nucleotides. Just to remind you, these videos are made from the book Medical Biochemistry, Molecules to Disease. If you need more details about any of these videos, you could please look at this book, which is available at payhip.com/ELD. So the chapter on nucleic acids and nucleotides is chapter 2. We are currently on this chapter that discusses water, inorganic chemicals, carbohydrates, nucleic acids, as well as lipids. It's always important to see the big picture. So one of the major functions of nucleotides is that they are chemicals that we use to store information. And so as you know, DNA has nucleotides in the form of information. Your G, C, and AT are basically parts of nucleotides. And this information is transcribed by a process called transcription into various forms of the other nucleic acid, RNA. Then these various RNAs that are used for protein synthesis undergo processing, and then they are exported out of the nucleus. And once they are in the cytoplasm, or this messenger RNA message is translated because now we have nucleotide language being converted into amino acid language. So we call this translation or protein synthesis, and the protein is made. The protein undergoes folding. Some proteins may be processed, and then the protein is transported to the right location in the cell where it's going to function, and your cell has all of these proteins, and these proteins help the cell carry out most of its cellular activities. And this is what we see in the 200 different types of cells that we have. And the activities of these cells is responsible for human anatomy and physiology in health. And in disease, it's responsible for pathology. So that's the big picture. This is where one of the important functions of nucleotides is. So what is a nucleotide? A nucleotide has 3 parts. A nucleobase, sometimes just called base, one or more phosphates, and a carbohydrate. The nucleobase, let's look at the nucleobase. Nucleobase could be a larger purine or a smaller pyrimidine. So the 2 major purine nucleobases are guanine and adenine, and the 3 important smaller pyrimidine nucleobases are cytosine, thymine, and uracil. So this is your G, this is your A, this is your C, your T, and your U. So what is so special in the chemistry of these nucleobases that they are very good as information chemicals? Why did nature, and in this case, since we are healthcare professionals and students, why does your body use these chemicals for information? So that's the question we need to answer. What's so special in the chemistry of these nucleobases that makes them very good molecules for carrying information? So one reason why nucleobases are used as information chemicals is because of their stability. These are very stable chemicals. Your G, your A, your C, your T, your U, these nucleobases are very stable, and that's because these are aromatic molecules. If you remember from your organic chemistry, aromatic chemicals are those that are cyclic, planar, fully conjugated with p orbitals at every ring atom, and the molecule has 4n plus 2 pi electrons. So these nucleobases satisfy all of these criteria. You can see the benzene ring-like structure in adenine. In this case, in guanine, if you write a resonance structure, you can see this ring here becoming something that looks like benzene. So if I draw this, here is your nitrogen, and this is a double bond now, and this is an O minus here, and then the rest of the structure. So you can see this ring, one of the resonance structure, looks like benzene too. And so similarly, you can do the same thing here, and you can see this will become a double bond, and this will become O minus, and that gives you a benzene ring structure in C, T, as well as U. So these nucleobases are very stable because of their aromatic nature. So being aromatic compounds are also very hydrophobic. So in nucleic acids, these bases, nucleobases, are found inside the DNA, or if the RNA that is folded, you'll see the nucleobases found inside, and therefore they are protected by the sugar phosphate that is found outside. So this information is well protected in nucleic acids. This is very critical information. As we saw, this is the information that makes all kinds of proteins that are necessary for our survival. There is a lot of information to store in DNA, your 46 chromosomes. A lot of information, and remember all this information is stored in a small part of a cell that we call the nucleus. So for that purpose, this information needs to be stored in a very compact way because there's very little space to store this information. And one of the nice things about these nucleobases is they are planar structures, okay? That's part of the aromatic nature, flat planar structures. So these information can be stored like pages in a book. The pages also are flat, two-dimensional structures. So we'll see that this ability to store this information as pages allows us to store a lot of information in our 46 chromosomes that gets packed into this small area that we call the nucleus of the cell. Another good thing about these nucleobases, when you look at the substituents such as amine, and here is your oxygens, these atoms can form hydrogen bonds with one another, and that helps a lot in the stability of DNA that we'll see in a future video on the nucleic acids. So that's another good thing about these information molecules, is that they allow nucleic acids to be more stable through hydrogen bonding. So the second part of nucleotide is phosphate. As you know, phosphates are negatively charged. So you see this negative charge here. They're hydrophilic, so they like water because they're negatively charged. And this phosphorus atom, because it's sp double bond O, those of you remember organic chemistry, these atoms are very sensitive to be attacked by what we call nucleophiles. Okay, so in your body there are a lot of nucleophiles, so these nucleophiles can attack these phosphoruses here. and allow certain chemical reactions to occur. The third part of nucleotide is the carbohydrate. And this carbohydrate is a monosaccharide. It could be either a ribose or, in case of RNA, and in case of DNA, it is a deoxyribose. So the D here stands for deoxy, and the R in RNA stands for ribo. Okay, so the sugar is very hydrophilic, likes water. So why ribose in RNA and why deoxyribose in DNA? So you can see the difference is this. At this position, deoxy doesn't have an oxygen, ribose has an oxygen. Well, this chemistry makes a huge difference because we want RNA to be unstable. We don't want RNA to live around in the cell for a long time, because the longer the RNA lives around, the more the proteins will be made. So we want RNA to be in the cell for a short time, carry out its function, and then get destroyed. So the presence of this 2-OH group here at position number 2 right here makes RNA very unstable. On the other hand, DNA is permanent information. We don't want DNA to be unstable. We want it to be very stable. So the absence of this oxygen here will allow DNA to be very stable, and we'll look at this under nucleic acids. Note that the numbering on the sugar, on the carbohydrate, is numbered with primes, C1 prime, C2 prime, and so on, while the numbering on your nucleobases do not have a prime. So whenever you see a prime, let's say 3 prime, we are always talking about the sugar and not the nucleobase. So since we have 5 different nucleobases and you have 2 different sugars, ribose and deoxyribose, and you could have 1, 2, or 3 phosphates attached, we have a diversity of nucleotides. So for example, here the nucleobase is A. So we start with an A. There are 3 phosphates, 3 triphosphates. So this structure here is adenosine triphosphate, or ATP. Now, if you had only 2 phosphates, you didn't have this phosphate here, only these 2 phosphates, then that would be adenosine diphosphate. And if you had only 1 phosphate, you didn't have these 2 here, then that structure would be adenosine monophosphate. Similarly, if instead of A you had a G here, a guanine, then you would have the corresponding GTP, GDP, and GMP. And if you have a C here, then you have CTP, CDP, CMP, and so on. If you have a T here, then you'll have TTP, TDP, and TMP. And if you have the U here, you'll have UTP, UDP, and UMP. So in this case, all the sugar is all ribose. But what if you had the deoxy form? Okay, what do you have a deoxy form? So if this sugar didn't have this OH, it's a deoxy, then you start your abbreviation with a little d. So if let's say there wasn't an oxygen here, then this structure would be deoxy. This is A, and there are 3 phosphates, that's TP, TP. So that would give you deoxyadenosine triphosphate, or DATP. In a similar fashion, if this was a G, then you would get your deoxy-GTP. If this was a C, you would get deoxy-CTP. And if this was a T, you would get deoxy-TTP. So for DNA synthesis, these are the 4 nucleotides we require. And for RNA synthesis, these are the 4 nucleotides that we need. Now, it's good to know that many times we use this abbreviation DNTP. In this case, N is any nucleobase, DNTP, deoxy-NTP. Or similarly, you can have NTP for the ribose form. So whenever we talk about monomers, such as carbohydrates, we said monosaccharides are monomers for carbohydrates. You can join monomers with another monomer to form dimers and then trimers and eventually polymers. So in carbohydrate, we saw how the formation of acetyl links two carbohydrates together. Now, what about nucleotides? How are nucleotides joined to one another? So the reaction to join two nucleotides is a reaction between the hydroxyl on position 3' of one nucleotide, let's say an ATP in this case. This is actually deoxy-ATP because there is no oxygen here. So this is a deoxy-ATP. Let's say it's reacting with a deoxy-TTP. That's a T here, and then again you have 3 phosphates, TTP. So you can see in this reaction, the OH attacks this phosphorus that is nearest the sugar. And in the process, it forms a phosphoester with this sugar here, and it forms another phosphoester with this sugar here. So since there are 2 phosphoesters, we call this whole linkage between this sugar and the next sugar, we call this phosphodiester, 2 esters, phosphodiester linkage. So this reaction between the 3' hydroxyl group of one nucleotide attacking the, what we call the alpha phosphorus, the phosphorus nearest the sugar of a second nucleotide, this reaction that then forms, that links the 2 nucleotides through their sugars via a phosphodiester bond. This is the reaction that we see when we synthesize DNA, and that's carried out by DNA polymerase, or when we synthesize RNA, and that is carried out by the enzyme RNA polymerase. So this is how 2 nucleotides are joined together, and A, for example, and a T is joined via their sugars by a phosphodiester. So just to summarize, 2 carbohydrates, monosaccharides, let's say a glucose with another glucose is joined by an acetyl, while 2 nucleotides, in this case an A and a T, is joined together by a phosphodiester bond. Another important concept, for example, when we talked about disaccharides and then polysaccharides, okay, so let's say a glucose joined to a galactose, we talked about one end being called the non-reducing end, and then the other end, we call that the reducing end. So similarly with a dinucleotide or trinucleotide or polynucleotide, such as DNA and RNA, we have 2 ends, 2 terminals, 2 ends. 2 ends of the molecule look different. So here is one end, this is called the 5' end, and this is called the 3' end. So the best way to recognize the end is look at the sugar, and if you see a 3' hydroxyl, then this is the 3' end, and if this is the 3' end, then the other end will be 5'. Okay, because you can see here, this is 1', 2', 3'. You can see here there is no hydroxyl group here. This is actually involved in phosphodiester. So this cannot be the 3' end. That means this must be the 5' end and this is the 3' end. Okay, so the best way to recognize a dinucleotide or trinucleotide or polynucleotide such as DNA is always try to look at this presence of the hydroxyl group at the 3'. That will give you the 3' end, and therefore the other end will be 5' end. So that's how the nucleotides are connected together in DNA or RNA via their sugars. So you can see the sugar phosphate. The sugars phosphate will be on the outside, while the bases, your nucleobases will be on the inside. So your sugar phosphate, here's the other strand of DNA, and the nucleobases are on the inside. Okay, so hopefully this will slowly help us understand the next video, which will be on nucleic acids. So what are the functions of nucleotides? See, in an earlier video, we mentioned about energy. Without energy, nothing is going to happen. So I would say this is the most important function. Nucleotides are not fuels. They are not fuels. But the energy that is released when these fuels are burned, especially in the presence of oxygen, that energy is stored as nucleotide energy, especially ATP and GTP. And one good thing about using nucleotides for energy storage is that thermodynamically, they are unstable. There's a lot of negative charges near one another that makes the molecule unstable. But that doesn't mean the nucleotide breaks down easily, because it is kinetically very stable. So very nice molecule for metabolism. You cannot break it down easily. You need probably an enzyme. In metabolism, you'll see you require enzymes or proteins to break down ATP or GTP. And it will be broken down because thermodynamically it's unstable, but it's kinetically stable. Probably the second most important function is these are the building blocks. Nucleotides are the building blocks of DNA and RNA. Okay, so they are the monomers for nucleic acids such as DNA and RNA. Another function of nucleotides is they are very important intracellular signals, especially cyclic AMP and cyclic GMP. And we shall talk about these under cell signaling. So nucleotides are very important intracellular signals. Another function, nucleotides such as UDP is attached to glucose to make glucose more reactive. And this is very important in glycogen synthesis. And glycogen synthesis is very important because glycogen is how we humans store glucose. And as we mentioned in an earlier video, glucose is the most important fuel for humans. Later in metabolism, we will talk about coenzymes, enzymes and coenzymes. And coenzymes are necessary for a number of enzymes to carry out their functions. And you'll see that nucleotides are part of many coenzymes. Okay, so nucleotides are an important component of many coenzymes that are very important in metabolism, which we'll talk about in a future video. So until the next video, goodbye for now. Hopefully we'll see you soon in the video on nucleic acids.